Today's lecture is all about amino acids, which are the monomers of proteins. So to start, we're going to talk about the general structure of any given amino acid. Then we're going to talk about how that structure can differ and lead to us having 20 different amino acids. Note, for the amino acids, you are going to want to have them memorized in terms of structure, full name, three-letter name, and single-letter name. Then we'll talk about how amino acids can combine to form polypeptide chains, and the opposite of that, how we can break down those chains. And we'll end off with some examples of protein modifications. So just as an overview, I wanted to go over how proteins are actually made in our bodies. So this is something that you've definitely gone over if you've taken like a genetics class or even in Gen Bio. So we start with the DNA in the nucleus, which would then be transcribed into messenger RNA. That messenger RNA then leaves the nucleus and it finds a ribosome on the rough ER in the cytoplasm. There it is translated into a polypeptide chain. And so in this picture they show, here's the polypeptide chain, and then they actually show it folding into a full-fledged three-dimensional structure, which would then be our protein. For us in this lecture, we're not too much worried about this folding stage. We're going to cover that more in the next one. So like I said, every amino acid basically follows the same general structure, and that's going to be a carbon attached to 4 groups. 3 of them are going to be the same in all 20. Of those, one is the amino group. On the other end, we have the carboxyl group, and then we have a hydrogen. The 4th group is known as the R group, and that's what we call the side chain. And basically, all of the 20 amino acids have different side chains. That's what makes them different. Another thing is that most of the 20 are actually chiral. So if you remember from Orgo, something that's chiral basically means if you took its structure and then you flipped it over and you got its mirror image, the two would not be superimposable. And we know that this happens when you have 4 different substituents. And so we already have 3 solid ones. And so then the 4th one is the only variable one. So as long as this R group is not one of these 3 groups, you would be chiral. The only one that is achiral is glycine, and we'll talk about it later. But basically glycine's side group is a hydrogen. That's it. And so that carbon would have 2 hydrogens, and it's no longer chiral. The other 19 of 20 amino acids, though, they are all chiral. Also, I just wanted to point out that in the next couple of slides, we're going to go over the structure of each of the 20, and they all are going to look a little bit different from this picture we have up here. So the difference is that the amino group is actually going to be protonated. So here it's an NH2, down here it's an NH3, and it's become positive. On the other end, the carboxyl group will actually be deprotonated, and so instead of being neutral charge, it's going to have a negative charge. And this is because this is what the structure looks like when it's at neutral pH, and we'll go over why also in a couple slides. So there are different types of amino acids. We have essential amino acids, which they're essential because basically our body can't produce them, and so we have to acquire them through our diet. And so there's 9 essential amino acids. They're also shown up here in this chart. And this actually shows you what foods offer you certain amino acids as well. On the other end of the spectrum, we have the non-essential amino acids. This is the smallest group, and those are actually the amino acids that your body can produce on its own. We don't necessarily need it through food. And then there's this middle ground group that basically sometimes our body is able to make it, but other times it's not. So the synthesis of those is kind of limited. We can also group amino acids based on stereochemistry. So like we mentioned in the last lecture, you can have a structure and then you can flip it and get its mirror image. So on the left one, I have the amino group on the left, and therefore this is alanine, which is a specific amino acid. So this version would be considered L-alanine, and then when we flip it and get the mirror image, now the amino group is on the right, and so that would be D-alanine. For our purposes in this course, talking about amino acids coming together to make proteins, those are all going to be in this stereochem form. The D version is really just for structural use in bacteria's cell walls, so we don't need to worry about those. So the 20 amino acids are generally categorized into 4 groups, and that has to do with the characteristics of each of their side groups. And so it's very helpful if we know our functional groups ahead of time and know which are polar, which are nonpolar, and then which can become charged, meaning they're acidic or basic. So just as a review, an hydroxyl group is polar, as well as a carbonyl, right? That ketone is going to make it polar, and then also a sulfhydryl. So any SH, that would be polar. On the other end, nonpolar things would just be essentially carbons and hydrogen. So here we have a methyl group, very nonpolar. And then in terms of acidic and basic, a carboxylic group, right? Carboxylic acid, super acidic. That's going to give off its H as an acid, and it's going to generally be negatively charged, those amino acid side groups. On the opposite side, we have amino groups, and these are able to accept an additional H. Right here we have NH2. We can get another one and become NH3 positive. And so these amino acids that are basic are going to generally be positively charged. And so this is how we're going to group them. And like we just said, these are the functional groups that are going to represent these categories of amino acids. Remember, acidic ones will be negatively charged, basic ones will be positively charged. We're first going to dive into the acidic amino acids. This is the smallest group. There's only actually 2 of them. For all of the amino acids, this is going to be the general structure that they're going to look like. So just to review, this is our carbon. We have the amino group. It's protonated because we're at a neutral pH. We have the carboxyl group, deprotonated again because we're at neutral pH, the hydrogen. This pink box is going to represent the side chain, so that's the part you're really focusing on. Also, all the amino acids, like I mentioned in the intro slide, they have a full name, a 3-letter name, and a single-letter name. And so you're going to want to have these memorized. I do think it's very helpful to come up with like silly mnemonics to relate either the name to the single letter or even to the structure. And so I'll share with you guys what my silly mnemonics are. You don't have to use them. If they help, I'm glad. If not, come up with your own. Yeah, so let's get started. So this is aspartic acid. You can see here the side group has a carboxylic group. It has been deprotonated because it's super acidic, and so it's negatively charged. The 3-letter is nice and straightforward. It's the first 3 letters. The single letter, however, is not what you would expect an A. It's actually a D. And the silly way that I remember that is if you just say aspartic acid with like a stress on the last syllable, right? Aspartic acid, dic, D, that's kind of how I remember it. The other amino acid that is acidic is glutamic acid, and it basically has the exact same structure. It just has an additional CH2. So here we only had, we had a 2-carbon chain. Now we have 3, but still a carboxylic at the end. It's donated, it's H, and has become negative. The 3-letter name is nice and straightforward, first 3 letters, and the single letter is E. The way that I remember this one is I think of like glue, like the sticky thing, and I just finish spelling the word between the 3-letter and the single letter. Also, one thing about the acidic amino acids, here they both are, they can also be known as aspartate and glutamate. So just keep that in mind. It can either be aspartic acid or aspartate, glutamic acid, or glutamate. Now we're going to go into the basic category. This one's only slightly bigger. There's 3 amino acids that are basic, and for the most part, like we mentioned, amino groups are the functional group we're going to be looking for. So basically any structure that has a plethora of ends, that's going to be basic. So here you can see we have a long chain, but look at all these ends, and so it's going to have a basic characteristic. So this is arginine, arg or arg for the first three, nice and straightforward. The single letter is an R. The way that I remember this is you're kind of saying the letter R when you say arginine. So that's how I remember that. This next one is lysine. Again, we have a nice long chain, and then it just has that protonated amino group at the end, so we definitely know that that's basic. Lysine's single letter is a little bit odd. It's a K. I have a silly anecdote to remember this. When I was younger, I had a friend named Kevin. He had lice, and so Kevin had lice, lysine, K. The last basic amino acid is histidine. This one actually has a ring that gets formed, but again we have these ends which are going to make it basic. And so histidine is nice and straightforward. Its three letters are the first, or its three-letter name is the first three letters, and the single letter is the first letter. And so now we've finished going over all of the acidic and basic ones. Because these are the only ones that will be charged, I like to remember them with an acronym, and I have it going across here. So I use the word DIRC. It's a made-up word, emphasis on the K because there's an H after it. And that'll be nice and helpful when we talk about the isoelectric point and the net charge on polypeptides, because these are the only side groups that are really going to affect those two things, and we'll talk about that later on, but just keep that in mind. DIRC, that's your acidic and basic amino acids. Now we're going to dive into the biggest category, and that is the nonpolar amino acids. So these guys are really just lots of carbons and hydrogens, of course, just in different like arrangements. So here I mentioned when we talked about the chirality of amino acids, this is glycine, the only one that is not chiral, and that's because its side group is a hydrogen. Now we have those two hydrogens, you're not chiral. The three-letter and single letter is nice and straightforward for this one. Next up we have alanine, so that's just one methyl group, that's it. The three-letter and single letter is also nice and straightforward. Next we have valine. Valine has a couple more methyl groups, and basically the way I remember this one, or how I relate the name overall, valine and V, to this structure is that you can actually see the V right here. Basically the side group is a V. In this case it's upside down, but depending on how you like draw out the amino acid, it could be up to the side, anything like that. Then we have leucine. This one, the structure is kind of like valine, but there is an additional CH2 here, and so it kind of makes it a Y, and that's how I remember it. Leucine kind of sounds like the name Lucy, which has a Y at the end, and so that's how I relate the structure to the name. The three-letter and single letter is nice and straightforward. So this is isoleucine, and iso means that it's an isomer of leucine, and so that's kind of how I remember it. If you know leucine's structure, basically what's happening is we have this 3-carbon chain, and then we had a methyl attached to the middle carbon. We're now going to move that to the first carbon. And so that's what isoleucine is. So we still have that 3-carbon chain that leucine had, except instead of having the methyl attached to the middle one, now it's attached to the first one. The 3-letter name for this one is the only thing that's a little bit odd. You would think it would be ISO, the first 3 letters, but that would just be saying iso, and they want to make it clear that it's isoleucine. So the I for iso, and then the LE to tell you it's an isomer of leucine. Next we have methionine. This one does have an S in it, however it's not a sulfhydryl group, meaning it's not an SH. The S is kind of sandwiched between these carbons, and so this one is actually nonpolar. The 3-letter and single letter is nice and straightforward. Yeah. Then we have phenylalanine. The name kind of tells you the structure. So if you remember, alanine was when we had just that one methyl coming off, so that one carbon. So this is alanine, but then with a phenyl group attached to it, and a phenyl group is just a benzene. Benzenes are, of course, super nonpolar, as long as they don't have any other substituents. The 3-letter name is nice and straightforward here. However, the single letter is not P, as you would presume. And the way I remember that is just, you're kind of saying, like, you're making the sound of an F when you say pH, right? Phenylalanine. So that's how I remember that. Then we have tryptophan. This one is arguably has like what I consider the biggest side group. It has 2 rings. We have a benzene attached to another smaller ring. This one does have an N, but don't think that this one is basic. We have 2 rings, which make it very nonpolar. The way that I remember the single letter W for this one's kind of silly, but like I said, this is like the biggest group, and so W, if you think about it, is like the fattest letter, and so you might trip over a W if you had to trip like finger quoted over a letter. Yeah, that's how I remember that. And also the 3-letter is not TRY, but it's TRP, but that's kind of nice and straightforward. Imagine saying trip. And then the last nonpolar amino acid is proline. So this is the only one that forms a ring, not only like within the side group, but it actually connects over to the amino group. This is the only one that does that. And it's actually special because it'll cause issues a lot in polypeptide chains. We can talk about it a little bit more, but just know that. The 3-letter and the single letter is nice and straightforward for this one. And so that finishes off the nonpolar amino acids. This is the largest group, so most of them are going to fit in here. Now we're going to dive into the last category, which are the polar amino acids. So first off, we have serine. Nice and straightforward. We have a hydroxyl group attached to a carbon, and so that's going to make it polar. The 3-letter and single letter is nice and straightforward. Then we have cysteine, which is actually very similar to serine. The only difference is instead of an OH, that hydroxyl, we have a sulfhydryl, and so it's an SH. Still polar, but yeah, so this is cysteine. The 3-letter and the single letter is the same, is straightforward. And the way that I relate these two is that I say that cysteine is serine's sister. Kind of like a play on the words, just so I know that their structures are similarly related, except this one is obviously going to be more S focused. Next we have threonine. This one also has a hydroxyl group, but instead of being attached instead of being the only substituent off of that C besides hydrogens, there is also another methyl group attached. The 3-letter and single letter is nice and straightforward for this one. Then we have tyrosine, which is essentially phenylalanine, except it has a hydroxyl group coming off the other end of that benzene, and that's going to make it polar. The single letter is the only one that's different, or not as straightforward for this one, but it's easy to remember because there's a Y in tyrosine, and so that's where the Y is coming from. And then we have the last 2 polar amino acids. This is asparagine, and the other one is glutamine. Note that they sound like aspartic acid and glutamic acid, and that's because they are essentially the polar counterparts of the acidic amino acids. So if you remember in aspartic acid, it basically had the same structure, except at the end we had a carboxylic group. So instead of having this NH2 over here, we had an O negative because it was acidic and had donated its hydrogen. So going from the acidic to the polar version, we're basically replacing that OH with an amino group, and that's just going to make this overall end polar, but not actually charged. And then the same thing for glutamine. this is the general structure for glutamic acid had this been a complete carboxylic group, but instead we've replaced the OH, or the O negative at neutral pH, with an amino group, and therefore it's just polar but not charged. Also, I forgot to talk about the 3-letter names. For asparagine, it's N, and for glutamine, it is Q. So these are a little bit odd. You might want to come up with a good mnemonic to remember those. And that finishes our entire chart. You definitely want to have this chart memorized. So you want to know the structure, be able to know which amino acid it is, and what category it belongs to. So I've been going on about how the amino group is protonated at neutral pH and the carboxyl group is deprotonated. So we're finally going to talk about why that's the case. And it essentially has to do completely with the relationship between the pH of the solution and the pKa of each group on the amino acid. So when we're comparing the two, obviously one will be, smaller than the other. And so the lower the pKa, the better the acid you are. So whichever one is lower, whether it be the pH of the solution or the pKa of the group, that thing is going to act as the acid. And so it's going to give up an H to the other one, which is acting as the base. So I know I have it written down here, but just to go over, so a carboxylic group generally has a pKa around 2, and then the amino group generally has a pKa of around 9. And that makes sense. Carboxylic is much more acidic, so it's going to have the lower pKa than the amino group. So let's talk about the first situation. So if our pH of our solution, in this case it's neutral, so it's going to be 7, is smaller than the pKa of our group. So that's going to occur on the amino end, right? This has a pKa of 9, pH is 7. So this is smaller than this, and therefore the solution is going to act as the acid, right? It has the lower pKa. And so it is actually going to give up an hydrogen, and it's going to head over to the amino group. And that's why this is generally protonated and has a positive charge. On the other end, our pKa of our carboxylic group is 2, and so the pH of our solution, 7, is greater than that pKa. Therefore, the actual carboxylic group is going to act as the acid and give off its H, and that's why we have an O negative here. It's given it off to the solution, which is acting as the base in this case. And so the outcome, our group is deprotonated. So that's what this chart kind of goes over. And this will be really helpful going forward, so you definitely want to have this concept down. Then down here, I have a list or a chart of all of the amino acids and then their respective pKs. So the first one, if you see, they all are around 2, and that's because this is the pKa of any amino acid's carboxylic group. The second pKa is generally around 9, and so that's going to be the pKa of any amino acid's amino group. And then there's a third column. Not all of the amino acids have it, and that's because the nonpolar and polar amino acids, their side groups aren't actually going to ever be charged. So only the acidic in red and the basic in blue, that's our DIRC, only they are going to have a third pKa, which would be of their side group, because those are the ones that may or may not get protonated or deprotonated depending on the pH of the solution. So now we're going to apply what we just learned about the relationship between the pKa of each group and the pH of the solution, and see how every amino acid can kind of have 3 different states. So we're going to keep track of the pH of the solution down here, and so it's going to start at 1 and we're going to increase it all the way to 14. So when it's super, super low, let's say at 1, the solution is obviously going to be acidic, right? The pH of the solution is less than the pKa of all of our groups because we're at 1, which is the lowest. Therefore, all of our groups will be deprotonated. Will be protonated. And so here we have the amino group is protonated, and that makes it positive, and our carboxylic group is protonated, which makes it neutral. And so this shows the fully protonated version of the amino acid, and it's going to have a net positive charge, right? When the carboxylic group is protonated, it just becomes neutral, but when the amino group is protonated, it gets that positive. So now we're going to raise our pH until we hit that first pKa of this amino acid, which is a 2, that's the lower one, which is the carboxylic group. So once we hit 2 and we're over that, now the pH of my solution is going to be greater than the pKa of my group, the carboxylic group. And so that group will then be deprotonated. So basically we're going to lose an H, which is coming from that carboxylic. And so the outcome is that that becomes negatively charged. The amino group has not been touched, right? The pKa over here is 9, and we're still somewhere between 2 and 9 for our solution. So that one's still protonated, this one's deprotonated. This is what we call a dipolar ion, which is basically, this is what we call a Zwitter ion, which is a dipolar ion, meaning it has positive and negative charges. The Zwitter ion will also balance out completely, and so it will be neutral. It has a charge of 0. Then we're going to do the same thing, and we're going to continue to increase our pH until we hit the next pKa, which is of the amino group around 9. So once I'm above 9, now the pH of my solution is greater than that second group on the amino acid, which is the amino group. And so we're going to lose another hydrogen, and now this becomes NH2, which is neutral. The carboxylic group is still deprotonated, and so that has a negative charge. So now overall, we are fully deprotonated, and we have a net negative charge. So that's how we're going to be walking through these. We're going to increase our pH and see how that increase affects each group. And then down here, I actually have, it's basically the same situation, just shown on a titration graph. So as you see, how we started with a low pH of 1 and increased it, that's what's happening here. We're going to increase our pH, right? We're going up. So in the beginning, when we were under our pKa 1 of 2, we were fully protonated, and so we were net positive. Then we continued to increase the pH until we hit 9, the pKa of our second group. And once we hit that point, we became negative. In between, we said we were neutral. And so there's actually something called the isoelectric point, which is actually the exact pH when your molecule, or in this case our amino acid, is in that Zwitter ionic state, meaning it is completely neutral. And so you can find that Pi, the isoelectric point, if we know the two pKa's that are right next to that, right, from going from positive 1 to 0, and then from 0 to negative 1, we can take those two, add them together, get the average, and that will tell us the exact Pi. So in this case, for our hypothetical amino group, which the R group doesn't get affected, the Pi would be somewhere around 5, 6, somewhere between there. And so now I just want to show you guys some comparisons. So the one we talked about was basically, like I said, the R group didn't affect anything because it did not have a pK 3 value of that side chain. And so, for example, this is glycine. This is the titration. The first pKa, that's the carboxylic group. The second pKa, that's the amino group. There's no third one, and so our Pi was the average of the two, again, somewhere around 5 and 6. Here we have the titration of lysine, which is basic. And so this is our first pKa around 2, that's the carboxylic. Then we have the one around 9, that's the amino. But then we have pKa 3, and that is actually the pKa group of the side chain, which is super basic because lysine is basic. And so that causes the isoelectric point to actually be a lot higher. Here it's 9.74, which is basic, and that makes sense because lysine is basic. Here, on the other hand, we have a acidic amino acid, so this is glutamic acid. We have the first pKa, that's the carboxylic, but we have another acidic pK, and that's the side chain. And then we have the amino group up here. So here, the overall pI is a lot lower, and that makes sense because glutamic acid is acidic. Okay, so now we're done talking about amino acids as single units. Now we're going to talk about how we can combine them to make those polypeptide chains. So here I have amino acid 1, and then here's amino acid 2. I have them both at neutral pH, so you do see the amino groups of each protonated and the carboxyl is deprotonated. The first one has some side chain, and the second one has another side chain. So when they get combined, what happens is basically we have water that leaves the system. And so that's going to be a condensation reaction because we're forming water. Where is that H2O coming from? Well, we have the O negative from the first one. That's going to come over here and attack that nitrogen and take off those H's. And they're going to come off, they're going to combine and come off. What's going to happen then is we're going to create where we had that bond going to the O, this C will then combine to this N. And that right there is your peptide bond. So then you just redraw everything, except the second one will kind of be in a flipped structure. So here I was going up, now I'm going to go down. But besides that, it's the same. We keep the amino group of the first one. It did, however, lose its carboxylic end, right? We still have the ketone, but now instead of having my O, or my hydroxyl group, I'm actually directly attached to the next amino acid. So it's going to lose that carboxylic group. The first one is kind of losing that protonated amino group, and it's getting attached, but it's maintaining its carboxylic at the end. And so this would be your full combined peptide chain. Again, they're combined with a peptide bond. Also, we talk about the front as the N-terminus, and then the back. So in this case, we only have 2, but imagine I had like 10 amino acids, only the last one's carboxylic group, we call that the C-terminus. Also in yellow, I have highlighted the backbone of any polypeptide chain. It's going to be a repeating pattern of NCC, then the next amino acid, NCC, and this would happen again and again for however many you had. Note, proteins are always synthesized from the N-terminus to the C-terminus, so it basically just means you're always adding a new one to the right end of the chain. We talked about this as condensation reaction, and then it's also good to note that this actually isn't thermodynamically favorable, and so when proteins are being translated, they're being coupled with ATP breakdown to actually force this to happen. And so here I'm just showing some more condensation reactions. We have 1, 2, 3, 4 different amino acids. The first 2 get added together, then that chain gets added to the next one, and then that chain will get added to the next one. And then you end up with a full-fledged polypeptide. Here's our N-terminus. Here's our C-terminus, and then it shows all the different R groups coming off. I do want to note there are some differences in the vocabulary we use to talk about polypeptide chains. So first off, if you have a polypeptide chain and you're talking about an individual amino acid in that chain, we call it a residue. So for example, here this is the aspartic acid residue within the chain. And then we have the difference between oligopeptide and polypeptide. Basically, oligopeptide is just a few amino acids combined together, and then a polypeptide would actually be if we had like more than 20. That's a full-fledged polypeptide. But don't worry about the difference, really. We're going to pretty much refer to everything as a polypeptide chain. Now we're going to look at the reverse of adding amino acids together, and we're going to take a polypeptide chain and we're actually going to break it. So this is a peptide bond, and we're going to break that peptide bond. We do that by combining the polypeptide chain with water, and that's actually going to act and break that bond for us. And then we're going to have the first amino acid. It's now regained its carboxylic end that it didn't have in the chain. And the rest of it, the first amino acid, the first residue in that chain, is going to regain its amino group. So this is known as a hydrolysis reaction, and that's because you need water as a reactant to break that bond. The enzymes that actually do this are called proteases, and so they are actually super specific. So they are only going to cleave the peptide bond next to certain amino acids. So for example, trypsin is a protease that's going to cleave the carboxylic side of basic residues only. And then we have chymotrypsin, which cleaves next to large hydrophobic residues. So if this was my polypeptide chain, we want to see where would these two enzymes break bonds. And we're going to see that trypsin is going to do it right here, right between the K and the G. And that's because K stands for lysine. So we're going to do it on the carboxylic end of lysine, meaning on the right of lysine. So we'd break it here, and then that would leave us with a chain of A, F, S, K, and then another chain, G, L. Chymotrypsin cleaves next to hydrophobic chains, and so that would happen between the F and the S, right? Phenylalanine is super hydrophobic, so we'd cleave it right there. Lastly, we're going to talk about post-translational modifications. So like I said, the polypeptide chain gets translated at the ribosome. After it becomes a polypeptide chain, sometimes we can have certain modifications. So we can either have amino acid side chains that are getting modified. So here, instead of it just being a normal serine, it's now been phosphorylated. There's a phosphate group. So this is phosphoserine. Here, proline, it's gained a hydroxyl group. Lysine, this one's gained an acetyl up here. And then glutamate or glutamic acid has gained a carboxylic. On the other hand, we can also update or modify the actual terminuses of the chain. So here we have the amino group getting modified, and then down here it shows the carboxylic group getting modified. And this can actually function in signaling pathways or, stabilizing structures or just playing roles in gene expression, which we're going to talk more about in another lecture, how these modifications can sometimes activate and deactivate proteins. And that's basically it for this lecture. So this is just kind of like a summary slide. Here we have amino acids that are composed of, we have the carboxylic, the amino, and the side chains. Then it goes on to categorize them into our 4 categories, and it talks about how some are, the acidic groups are deprotonated, basic ones are positive, things like that.